malignant pleural. So what is the pleural? What is mesothelioma? So on the left over here, you'll see there's a normal lung, and surrounding the outside of the lung is the pleura. The pleura is like a negative space filled with a lubricant that lets you breathe without any friction. Now, when mesothelioma occurs, it forms along the visceral pleura, which is the inside layer of that sac that covers your lung. It creates this diffuse-like tumor, unlike those ball-like tumors we're used to seeing in solid cancers, across the visceral pleura, and there's also the formation of the pleural effusion within the lung. So mesothelioma is primarily caused by exposure to asbestos. Now, asbestos has a very, very long history within Canada due to the fact that we were once a leading global exporter of what was called Canadian gold. It was largely mined in Quebec. There were many mines across Quebec, and one town was even called Asbestos for quite a while until a recent name change due to the negative connotations of asbestos today. It is still currently a leading cause of death in the workplace. Well, but mesothelioma is still a leading cause of death in the workplace due to the fact that many of our buildings still contain asbestos to this day. So there still is exposure to it on the daily work site. And the CDC has even stated that everyone is exposed to asbestos at some point in their lifetime. Now, the latency period for mesothelioma symptoms is 20 to 30 years, and they are non-specific symptoms that occur without any real emphasis on what it could be, which often leads to a late-stage diagnosis. Now, all this together with the 30-year latency, the non-specific symptoms and late-stage diagnosis leads to very limited treatment options. Now, all this together leads to a very highly deadly cancer with a 5-year survival rate of about 9% as of this year. Now, this has improved in the last few years. I would say 2 years ago, the 5-year survival rate was still around 5%. It is currently as deadly as pancreatic cancer in Canada. And in 2019, we saw about 460 diagnoses. Till today, we see about until 2022, when we saw 472 deaths. So what actually causes tumors? How does the asbestos lead to tumorigenesis? Well, when you inhale asbestos, they are these tiny little fibers, as seen here on the bottom. They're about 10 to 100 micrometers to 1 to 4. They're inhaled and they go down to the bottom of your lung and fall into your alveolar sacs, where they then are interact with your alveolar macrophages, which try to engulf them. And in the attempt to engulf the asbestos, they're unable to clear them, which then leads to the biomineralization of the asbestos, which is seen in orange there. And this leads to a high iron content around that part of the lung. Then over time, which largely explains the 20 to 30-year latency that we see with this disease, these asbestos fibers then travel further and deeper into your tissues through all the layers to then eventually get to your pleura, where they then settle and then impale into the mesothelial cells that make up your pleura. Once they've impaled the mesothelial cells, they then cause cell damage, which leads to the promotion of macrophages to come to the site and try and clear the asbestos. And they are also unable to clear the asbestos, which then leads to the promotion of inflammatory mediators and results in DNA damage. All this together then leads to tumor formation. So there are 3 distinct histological subtypes of mesothelioma. The most common type is the epithelioid subtype, you see at the top there. And this makes up about 60 to 80% of all cases, and it has a more organized-like structure within its histology. The next most common type is the sarcomatoid mesothelioma, and this has a much more disorganized type of histology and makes up about 20% of all cases. Between the two, we see the biphasic subtype, which is a mix of both the bottom and the top and bottom subtypes within one, and they tend to show characteristics of both diseases. And from this point on, I will be referring to the bottom two as the non-epithelioid subtype and the top one as the epithelioid subtype. So we're seeing distinct histology, which implies a distinct biology, which then leads to very distinct survival curves that we see here. So if you look at the top right curve in red, that's the epithelioid subtype. They have about a 9-month survival over the curve in blue, which is a sarcomoid subtype underneath. Now the red, at about 50% survival, we still see that happening at around 14 months, and in blue for the sarcomoid subtype, that occurs at about 5 months. These are very short periods of survival. This is an incredibly deadly disease, and while many will refer to the epithelioid subtype as having a better prognosis, that prognosis is still incredibly deadly. So what might be causing this? Why are we seeing such a distinct survival between these two subtypes? that could largely be explained by the very distinct tumor immune microenvironments that we're seeing. So over here on the left, we look at the epithelioid subtype first. They have a much more what they call innate-like immunity within their tumor microenvironment. They're made up of not many T cells, but we do see some Treg cells in there, a few natural killer cells, and some MDSCs. And if we shift over to the sarcomoid subtype, we see much more adaptive-like features within their tumor immune microenvironment, which is largely represented by the fact that they contain PD-L1 and PD-1 biomarkers, which indicate that at some point within this tumor, there has been an immune response that has occurred that has then been stopped by the expression of PD-1 or PD-L1. So up to this point, there haven't been very many great treatment options for mesothelioma. For the most historic treatment option was surgery with chemotherapy, but thanks to the rise of immunotherapy, we're now seeing that there is benefit when treating patients with mesothelioma with immunotherapy. So this is checkmate for 743, which is now the first-line treatment for mesothelioma. And as you can see here, when you treat patients with anti-PD-1 and CTLA-4, there's somewhat of an improvement in survival in the blue line, which is the immunotherapy group over time. However, if we break it down to the two subtypes, epithelioid and non-epithelioid, we see that the benefit for survival is largely maintained within the non-epithelioid subtype, while our most common subtype, the epithelioid subtype, sees almost no benefit at all. So while this is a great immunotherapy for about 20% of our patients, it's not addressing the larger problem. So our lab is incredibly interested in understanding why this is the case. We have developed two panels to address this, two multiplex immunofluorescent panels to address this. In our first panel, called the Axis panel, we have molecules that are known to be markers of response to immunotherapy. While in our second panel, we have markers, our targetable molecules panel, we have markers that are currently in clinical development and suggest that they may provide a therapeutic benefit to the patient when drugged. So why are we interested in these targetable biomarkers with mesothelioma? Well, mesothelioma expresses a whole bunch of those markers I just showed on the last slide. Specifically, they express TIGIT and TIM3 on the T cell, as well as there's this expression of this molecule VISTA on the tumor cell and on the T cell. Now, why is VISTA so interesting within a mesothelioma case and not the other markers I've shown before? Well, VISTA has the highest expression within mesothelioma across all solid tumors to date. So when looking at the TCGA database, they found that VISTA had the highest expression level across all solid tumors. And this is shown here in the all-in-PM bar graph on the left. And then if you go through the different subtypes, you see there's a decrease in the VISTA expression as we move from epithelioid to sarcomatoid. Now, this was interesting when thinking back to the immunotherapy results, where we saw that the sarcomatoid subtype was not, the epithelioid subtype was not responding to anti-PD-1 therapy. And if you look over in figure B there, those are IHCs staining for VISTA on the top, and you see a stark decrease in the stain, which is in brown, as we move through the subtypes, and then PD-L1 on the bottom, which is an increase, which confirms what we see in A. Now, this is at the protein level. If we look even further into the mRNA level, we also see the same thing occur again, where there's VISTA expression within the epithelioid subtype is way above that we see within the sarcomatoid and biphasic types. Now, where is VISTA being expressed within the cancer? Up to this point, we know that VISTA is currently expressed on the tumor cells, as seen in red here, the red arrows, and then also on immune cells, as seen in black, and the black arrows. So what is VISTA at a protein level? Well, VISTA is a PD-1 homolog, and it binds to two very well-established receptors, such as PSGL1, which is a well-known p-selectin receptor, and then also VSIG3. Now, VISTA is a pH-sensitive protein, and it will bind to each of these receptors at different points, depending on its pH that it's in. So as we know, tumors tend to have a very acidic-like pH, and the receptor that it binds to within this pH is PSGL1 seen there. So I would expect within my project that that's what it's binding to, and it's often expressed on endothelial cells, among other things. Okay, and what else do we know about VISTA? Well, we also know that VISTA is very important on T cells for maintaining a high threshold for T cell activation, ensuring that within our system, we maintain tolerance to self-antigens. So VISTA, there is this mouse model that was done recently in 2020, where they looked at VISTA and they found that when there was a knockout of VISTA within the mice shifted from a more self-tolerant phenotype to a loss of tolerance and very immune-like, autoimmune-like features. Many of the mice end up developing lupus and other autoimmune diseases. And what they then overall just came to conclude was that VISTA was the first in the series of T cell activation checkpoint molecules. So VISTA is the first one there to make sure that when our T cells come into contact with their antigen of interest, there's a high enough threshold that we are not self-reacting to our own antigens. And then CTLA-4 and PD-1 have other downstream factors after this initial activation has occurred. So what is my hypothesis? Well, my hypothesis is that the expression of VISTA in the tumor immune microenvironment is specific to CD8 T cells and tumor cells can be a predictive biomarker for negative treatment outcomes in epithelioid malignant pleural mesothelioma. And how am I going to study this? Well, we're lucky enough at the Control Lab to have access to a whole bunch of clinical samples from the N227 clinical trial, which was performed with the Canadian Cancer Trials Group at KGH. And what they did is they looked to see if there was any clinical benefit to treating mesothelioma patients with anti-PD-1 and chemotherapy. And what they found were results that were very similar to those seen in the Checkmate 743 trial, where the non-epithelioid subtype over here on the left had a result, while the epithelioid subtype had almost no benefit to immunotherapy. And what will I have access to? Well, I'll have access to about 300 pre-treatment samples from the phase 2 and phase 3 clinical trials. This is a very remarkable number, as the TCGA, which is where most mesothelioma work is currently done, only has 74 cases. And then with that, we're really hoping in the lab as a whole to come up to map a very significant amount of mesothelioma cases and then link that back to biomarker development. So what we do in our lab is multiplex immunofluorescence. And the way that we do that is we start with a wax embedded tissue, as seen here, paraffin embedded. And then we slice that up and we stain it with our automated stainer, the Bond Leica. And then we move on and we image with the vector Polaris, and we analyze our data within Inform. So this is a pipeline that we repeat iteratively over and over and over again until we find that the stain that we have is representative of what's happening biologically. So there are many different ways to stain tissue, as I'm sure you've all heard. And the way that we do ours is through TSA Opal technology. So the way that this technology works is you have the primary antibody come in and bind to its epitope of interest, followed by the secondary antibody, which is bound to enzyme, which then catalyzes the reaction of the fluorophore onto the tissue, where it covalently binds to the site of the antigen. Then what we can do is we can wash off the primary and secondary antibody and stain again with a new fluorescent molecule. And we can repeat this over and over and over again till we can get up to about 6 to 8 markers on the same slide. And this is great because then we can image and we can then unmix them. But that's only 6 to 8 markers. How much can we really figure out with that? We're looking for new ways to expand our current data to get more information out of our one slide. So we've now gone on to look for new technology, and we found technology in the States from this company called AltaView. And the way that they are different from what we currently do is that their fluorophore does not covalently bind to the tissue. Instead, it binds to the primary antibody. And this is beneficial to us because we can go in and stain our tissues initially with this technology, and then wash off the primary antibodies and restain again with our optimized TSA Opal panels. This will then expand our 6 to 8 markers to about 10 to 12. Giving us that much more information to work with. So what are my aims with this project? Well, my first aim with this project is to optimize a pan-membrane stain to improve single cell segmentation and downstream single cell phenotyping within this phenotyping analysis side of this pipeline. And then once that is completed, and it almost is, I will then move on to increase the number of antigens we can stain for by validating the addition of AltaView technology to our TSA Opal panels. And then finally, I will go on to stain the 300 preclinical samples from the N22 study, and then analyze them using Astropath technology and elucidate if VISTA spatial expression and VISTA cell-specific expression has a relation to overall survival. So why am I focusing on the cell segmentation, and why do I have to optimize this segmentation stain? Well, as you can see here in Inform, which is the typical software that is used today to segment cells when looking at multiplex images, you'll see that in the top left there, the cell there has been over-segmented. We're seeing 4 when there's really only 1. And this is a current issue that a lot within the analysis pipeline is dealing with. Well, Astropath has been able to develop a new way to go about cell segmentation. This involves machine learning, where the machine will go in and look for big cells and small cells, separate them out, and then segment them from there. Because the issue that they found was that in Inform, they're unable to tell the difference between big cells and small cells, and will try and segment big cells like small cells or small cells like big cells, and can't differentiate between the two. So what AstroPath was able to do was create the differentiation, and then they decreased over segmentation by 25%. And what was I asked to do to optimize this? Well, the original stain seen over here on the left has a lot of background, as seen in the red, which made it a little bit difficult to figure out where the cell membrane is. It's not that high contrast. I was asked to remove the background, which I did, as seen over there on the right. There's a lot less background, as you can see in the red there, and there's higher contrast of the cell membrane. Now, I'm just gonna make a point here that what I'm aiming for with this project is to have a highly specific membrane stain, while when I move on later to talk about our panels, we're looking for highly, and the word sensitive, sorry, everyone, stains. So how did I go about getting the pan-membrane stain? Well, initially I was given 3 different antibodies, so the stain is made up of a cocktail of CD44, CD45, and the sodium, potassium, ATPase. I then went in and played around with the brightness through TSA titration, where I played around with our fluorophores and diluted them or concentrated them depending on how much contrast I was getting and how much background I was losing or gaining. And then finally, I validated them with the segmentation performance to see if it was improving. I did do this in Inform, so I ended up building 2 different algorithms, one for small cells and one for big cells, and looked at them individually. And then finally, I hope to move on and validate it with our AIM2 in a whole multiplex panel, and then move on and validate it within AstroPath itself. So once that's all done, and maybe at the same time, I will then move on to validate our AltaView in situ-plex technology along with our targetable molecules panel. Now, the benefit with the AltaView in situ-plex technology is we'll add on 4 more markers, and these 4 markers will be cell lineage specific, which means that I'll be able to tell on the slide exactly which cell types these other 7 markers are associated with. And from there, we can then figure out if VISTA is expressed on T cells, if it's expressed on tumors, and then there may be other cell types it's expressed on, which I may not pick up here, but I'll be able to tell and then can go in and build more panels to look at that deeper. And once that's all done and I'm able to move on and then stain my clinical samples, I will stain the clinical samples with the 2 panels, move on to whole slide scanning, and then do my analysis within AstroPath. And then what do I expect to see? I expect from the targetable molecules panel specifically to see a high VISTA expression on the epithelioid tumor cells, as I saw in the readings, for there to be a low VISTA expression in the non-epithelioid subtype, then for there to also be expression on T cells within the epithelioid, and then not so much in the non-epithelioid subtype. And then finally, I want to have a look and see if there's a correlation between the VISTA expression and FOXP3 expression, and if FOXP3, which is a known immunosuppressive marker, is also increases with VISTA expression. And then finally, what do I hope to gain from the greater overall project? Well, I'd like to understand if the myeloid cell markers on the axis panel correlate at all with the VISTA cell markers on the targetable molecules panel, and what their relationship is with overall survival. And that's everything. Thank you, guys.
All right. Hi, everyone. My name is Julie, and today I'll be presenting my proposal entitled Investigating the Mechanisms of RET-Mediated Invasion in Pancreatic Ductal Adenocarcinoma. So pancreatic ductal adenocarcinoma is a mouthful. I'll be referring to it as PDAC from now on. It's the most common type of pancreatic cancer, and it makes up approximately 90% of cases. And it's different from other pancreatic cancers because I'm just getting a laser pointer here. Can you see that? Yeah, perfect. It originates in the epithelial cells lining the ducts of the pancreas. So this makes this type of cancer exocrine in nature versus other pancreatic cancers, which are endocrine or neuroendocrine in nature, originating from hormone-secreting cells like those of the islets of Langerhans. So the majority of cases of PDAC originate at the head of the pancreas due to concentrated tissue, ductal tissue specifically. And as we all know, pancreatic cancer is a highly lethal disease. So some background about the state of PDAC in Canada. In 2023, it was projected to be the 9th most newly diagnosed cancer case, so incidence. in combined male and females, but then this jumps drastically to the third leading cause of cancer-related death in combined sexes. So this is because pancreatic cancer, specifically PDAC, has a 5-year survival rate of less than 10%, which apparently, according to Julia, is very similar to mesothelioma. And this is amongst the lowest of all cancer types, this 5-year survival rate. And unfortunately, there's been minimal progress over the past 20 years in trying to increase the survival rate. Pancreatic cancer, specifically PDAC, is resistant to a lot of common cancer therapies, including chemotherapies and radiation. So this points to the need for research into novel therapeutic targets or novel therapies to target this cancer. So why is pancreatic cancer so lethal? It's multifactorial in nature, the first being that there are minimal early onset symptoms. So a lot of individuals who have this disease don't know they have it until it's progressed. So this leads to late stage detection, and approximately 80% of cases already exhibit regional spread or organ metastases upon diagnosis. So this cancer is able to disseminate early and create micrometastases. So even when tumors are deemed resectable, a lot of the times the surgeons can't actually see a lot of the micrometastases. And it's able to spread through ways such as vascular invasion, lymph node spread, but then also a special way that is highly prevalent in a PDAC is called perineural invasion. So yes, again, all of these factors leads to a high recurrence rate after surgery and contribute to the low 5-year survival rate. So what is perineural invasion? It's when cancer cells infiltrate and disseminate along peripheral nerves, and it's present in approximately 70 to 90% of cases of PDAC, so a high proportion. And it contributes to PDAC lethality and morbidity. So first, PNI with PDAC is associated with decreased response to treatments, but then also another facet of this is that it causes extreme abdominal pain and nerve pain in these patients. So with the limited amount of time that they have to live, unfortunately, it is also extremely painful. So this means that we should identify, or points to the need to identify molecular therapeutic targets that can either slow or halt the progression of PNI and PDAC. To accomplish this, we first need to take a deeper dive into the molecular mechanisms that underlie PNI. So perineural invasion is a complex process that involves reciprocal exchange of signals and chemoattractants, both between the tumor cells and the nerves themselves. So another lab mate of mine, Larissa, is going into more depth on this reciprocal interaction. But what's important for my project is that one of the chemoattractants is called glial-derived neurotrophic factor, GDNF, and other family ligands, so GFLs in total, and their co-receptors. Specifically, these ligands these neurotrophic factors are the ligand for the RET receptor, which I'm sure a lot of you have heard about. That's the main focus of our lab. So the RET receptor rearranged during transfection. It's a proto-oncogene that encodes a receptor tyrosine kinase, so it's located on the plasma membrane normally in normal physiology, and it's important for development and maintenance of the enteric nervous system, the kidneys, and other neural crest-derived tissue. So RET becomes activated upon binding of GFLs and the co-receptors GFR alpha. Specifically here we have GDNF and GFR alpha 1. This causes receptor dimerization, autophosphorylation of intracellular residues, and then here several intracellular proteins can actually interact with RET to then mediate downstream signaling pathways. Here I show Src kinase, and these activate pathways such as the PI3K-AKT pathway, which is important for proliferation, but also in cancer is associated with invasion and metastases. So as I mentioned previously, RET can act as an oncogene. So the first way is when there are mutations that result in a gain of function, so increased signaling through RET. And the second way being increased signaling through an increase in wild type expression, so in tissues that don't usually express RET or an elevated expression. This is the latter of which is the case in RET and PDAC. So RET is expressed in approximately 40 to 65% cases, where elevated expression has actually been shown to correlate with higher grade tumors and poorer prognoses. So this makes us ask the question, does RET activity perhaps promote PDAC progression and aggressive phenotypes? And if so, how? A previous PhD student from the Malignant lab, Dr. Eric Lian, addressed these questions by exploring the role of RET in PDAC cell invasion and other functions into using in vitro models. So first he used wild-type PDAC cells, which express RET endogenously, and then he compared them to PDAC cells, which he modified using shRNA knockdowns. So he knocked down RET expression in these cells. Ultimately, he found that RET activation was required for cell polarization. So here on the left, we can see the mock knockdowns, which express RET, and they have stress fiber organization and polarization towards the source of GDNF, which is the ligand for RET. And then on the right here, we have RET knockdowns and a more dispersed and unorganized actin pattern. And then he also found that when looking at cell invasion, cells expressing RET demonstrated increased ability to invade into a surrounding collagen matrix compared to those that had a knockdown of RET. And then finally, there was also an increase in a proteolytic ability of the extensions in the cells that express RET. So here we have a red fluorescent gel, and the areas void of fluorescent are indicative of proteolytic structures that are able to degrade those areas. And then here with the RET knockdowns on the right, we can see that there's a lack of those areas void, or those void areas. So this points to perhaps RET being involved with the creation or promotion of proteolytic structures. And the one I'm going to look at specifically are called invatopodia. So in here, Dr. Eric Lian then looked at colocalization with RET and a specific known invatopodia marker in pancreatic cancer cells called TKS5. And he found that they were colocalized at the leading edge of pancreatic cancer cells. So this makes us wonder, does RET-mediated invasiveness in PDAC, and it may involve TKS5 activity and the promotion of invatopodia formation. Before I move on, I'm going to quickly talk about invatopodia. So they're actin-rich cell extensions. They're proteolytically active, so they degrade the extracellular matrix through the release of matrix metalloproteinases, or MMPs for short. And they're initiated usually by pro-invasive signals from receptor tyrosine kinases. In the past, it's been shown EGFR MET signaling, and now we're going to look at RET signaling. So TKS5 specifically is an adapter scaffolding protein that has been shown to play an important role in the initiation of invatopodia, specifically the formation of complexes, protein complexes, with proteins that are important for the maturation and the building of the actin. And interestingly for us, TKS5 is phosphorylated in a substrate of Src kinase. And previously it's been shown that Src becomes activated and interacts with RET. So this brings me to my hypothesis, which is that RET activation results in Src-dependent recruitment of TKS5, which in turn promotes the formation of invatopodia structures necessary for PDAC cell invasion. To address this hypothesis, I came up with 3 experimental aims. The first is to expand off of Eric's work and explore RET's contribution to PDAC cell polarization, motility, and invasion, but this time with the addition of RET-specific inhibitors, which have now been used in the clinic for other types of cancer. And then I'm going to look at and characterize the nature of RET-mediated cell extensions. So perhaps they are proteolytically active, but are they invatopodia? Do they colocalize with invatopodia markers? And then finally investigate the role of TKS5 and Src in RET-mediated extension formation. To address all of these aims, I will be using in vitro models, specifically using 2 different PDAC cell types, which both endogenously express RET. So we have PANC1 cells here, which express relatively more than MIA-PaCa2 cells on the right here. And there's distinct phenotypic and morphological differences between the cells. MIA-PaCa cells are more motile, while PANC cells are flatter. And then we also generated CRISPR-Cas9 gene knockouts, or RET knockout. Thank you, Dr. Brandy Hyman. And then TKS5 knockouts, which I've been working on for the past 2 months or so. And to first characterize these cells, I'm just trying to figure out how to get rid of the laser point. There we go. I'm going to look at RET-mediated signaling in these cell lines. So over a GDNF time course is their downstream protein activation with proteins that are involved with migration invasion, like TKS5, Src, and AKT. A little bit more about the PANC TKS5 CRISPR knockout cells, because I've been spending a lot of time on them. We transfected PANC cells with 2 different DNA constructs which had guide RNA targeting 2 different regions of the TKS5 gene, because there is a long isoform and a short isoform. So one construct targets just the long isoform, while the other targets an area downstream here that is in both the long and short. And as of last week, we have one pretty successful TKS5 knockout, which is exciting. And this was done with the help of an SLC student named Dax. So back to my aims. First, I'm going to look at PDAC cell polarization and RET. So cell polarization is a key step that precedes motility, specifically directional motility. And previously, Dr. Leanne showed that RET is required for stress fiber formation in these cells, but what's unknown is the effect of RET abolishment, so complete RET knockout, and RET-specific inhibitors on extension formation and polarization. Next, I looked at cell motility and migration, which I actually have some preliminary data for you here. So we first looked at MIA-PACA2 single cell motility over a 12-hour time frame, and it was found that the wild-type cells, so express RET, were far more motile than the RET knockout cells, both plus and minus GDNF, and then the addition of RET ligand GDNF also increased motility. And then these are individual paths of the cells. So MIA-PACA wild types plus G, wild types minus G, and then the knockouts. It's more sporadic movement here with the wild types plus G. The next steps would be to add a treatment with a RET-specific inhibitor to see if that abolishes or brings down the motility, as well as to add a RET or to do a RET add back in the RET knockouts to see if that recovers the phenotype. I'd also like to do directional cell migration towards GDNF and then a collective cell migration with wound healing. Next, we're going to look at RET activation in PDAC cell invasion using transwell invasion assays. So previously, Dr. Leanne found that RET activation was required for invasion towards the source of GDNF, but what's unknown again is the RET-specific inhibition and RET knockout. So using the transwell assay, we will seed the PDAC cells and then in a condition with either minus or positive or minus GDNF and watch and quantify invasion through a matrigel, where we'll also have a positive GDNF secreting control, so smooth muscle cells, which secrete GDNF, and then a negative control. And then we can also look at the effect of inhibitors on this process, like SRC inhibitors, RET inhibitors, and then Rho family GTPase inhibitors, which are important for cell polarization. For my second aim, we're going to look at RET invatopodia formation. So we can do this using in situ zymography, extending off of what Eric did in the past, Dr. Leanne did in the past. We're going to use something called either dye quenching or FIT-C collagen, where it quantifies fluorescent collagen degradation. So as we can see here, this was done in gelatin in another study, but you can see the dips in gelatin fluorescence and the increase in fluorescence of antibodies that are important for, or markers, antibodies that are important markers for invatopodia like cortactin and actin. So we're going to look at and see if RET colocalizes with invatopodia markers at these sites of degradation. And then we can also supplement this with gel zymography looking at MMP 2 and 9, which are important for invatopodia formation or invatopodia maturation, and quantify the proteolytic activity of these cells. So we can ask the question, does RET accumulate at areas of degradation or with other invatopodia markers? And does RET inhibition and knockout affect MMP release and degradation of these matrices? Finally, we're going to tease apart the RET and TKS5 interaction in PDAC cells. So previously, Dr. Leanne found that there was colocalization of RET and TKS5 at leading edges of PDAC cells, but it's important to note that colocalization is not the same thing as a direct interaction. So you can use something called a proximity ligation assay, which detects a direct interaction between two proteins that's less than 40 nanometers. So we're going to see whether TKS5 directly interacts with RET, and then we can also see a TKS5 and SRC interaction, and if that increases upon GDNF treatment. We'll also hopefully use total internal reflection fluorescence microscopy, or TIRF, and this assesses colocalization, but specifically at the plasma membrane. So here you can plate the cells on a collagen-coated cover slip, and then you can use a laser that comes in at an angle, and it only fluoresces the fluorophores that are near the cell membrane. And then we can look at the effects of SRC kinase inhibitors on this process. So if we add a SRC kinase inhibitor, does that abolish the colocalization of RET with TKS5 at the cell membrane? And finally, I'd like to repeat the functional assays in the TKS5 knockout cells. So I will just use those as another condition alongside the RET knockout ones. And we expect to see that knocking out TKS5 will impair invasion in these cells. Whether or not it will do anything with cell polarization and motility, however, we'll have to find out. So just some quick significance of my work. Hopefully will implicate RET inhibitors, which are, I said previously, are used clinically for certain RET expressing cancers like non-small cell lung cancer. And it will hopefully implicate these inhibitors or potential treatments for PNI and PDAC specifically, and create a foundation for checking this in preclinical models. It will also establish mechanisms of RET-mediated invasion in PDAC, which is important because if these small molecule inhibitors for RET end up, there's a huge problem with resistance to small molecule inhibitors. So if we identify alternative targets in this pathway, that can perhaps reveal other ways of abolishing this process, or at least impeding this process for a small amount of time, hopefully. And then finally, just a broader understanding of oncogene-mediated invasion in general and metastasis. And this could be transferable perhaps to other tumor types that also exhibit increased receptor tyrosine kinase activation. I'd like to say thank you to my supervisor, Dr. Lois Mulligan, all my committee members, and then my lab members, and Dr. Brandy Hyman, a research associate. Thank you.
Right. Thanks, Julie. Questions for Julie?
Yeah, you have a question? Thank you.
Just curious, like you mentioned SRC, is there like any like thoughts of using SRC